Cells of the immune system, natural killer cells. In about 1975, Rolf Kessling was studying tumor cells and found that there was a cell which could kill tumor cells, and he found that it was a highly regulated cell, genetically regulated cell, and therefore he called it as the natural killer cells. So this session is going to be giving you a small or a basic overview of natural killer cells. So let us look at the learning outcomes of this session. Found in peripheral blood with no characteristic surface markers, as is observed in B cells and T cells. So you know that B cells have a characteristic B cell receptor and would also have the B7, etc., while T cells could have CD4 or CD8 plus. So there are very characteristic surface markers present on them. One doesn't find highly characteristic surface markers on the natural killer cells, although there are certain surface molecules which are considered to be present in the killer cells, but more is to be known to find out whether those molecules are only specifically confined to the natural killer cells. So originally, when they did not find such characteristic surface markers on the lymphocyte, they termed it as null cells. But now it is more known as the natural killer cells. So they have established its role mainly in the innate immunity, as the response is not highly specific and they are able to produce cytokines and effector molecules. They have found to be effective for its antiviral as well as anti-tumor activities, which is due to the surface molecules present on its surface. NK cells can either activate or inhibit based on the specific type of receptors present on the NK cells and the signal transduction that happens thereof. So when you look at natural killer cells, therefore, what is important to note is this, that the natural killer cells are also developed in the bone marrow, and in the bone marrow, the stromal cells give rise to several growth factors, which eventually lead to formation of the common lymphoid progenitor, and this common lymphoid progenitor is responsible for giving rise to or differentiating into natural killer progenitor cells, and these natural killer progenitor cells then differentiate into what is called as mature natural killer cells, and these mature natural killer cells then move into circulation in blood vessels. So here effectively what has been observed, that common lymphoid progenitors and the CAR cells play an important role in the development of the natural killer cell. So the progenitor cells which give rise to natural killer cells, about 5 to 10% of the total cells in the blood or total cells of the lymphocytes that circulate are natural killer cells. CXCL12 that is released by the CAR cell and the IL-17 that is released by the stromal cells are able to lead to differentiation of the natural killer progenitors into mature natural killer cells. Now, in the bone marrow itself, as you can see, that the hematopoietic stem cells, first from the common lymphoid progenitor, and this common lymphoid progenitor then releases IL-2. This is actually CD122, that is a surface receptor that is present, and this is responsible for giving rise to an irreversible development from CLP to natural killer cells. So presence of this is extremely important. So as you can see, as it moves forward, with every development stage, what you find, this is the initial natural killer cell leading finally to mature natural killer cell. And when it goes from the A stage to the F stage, one finds that several surface markers or surface proteins are getting expressed. No T cell receptor or no CD3 complex has been observed in the natural killer cells, although the natural killer cells are more analogous to the T lymphocytes rather than the B lymphocytes. Uh, Nonetheless, they have several cytokine receptors, and because they have the cytokine receptors, they are able to, accept signals from outside, from different cell types of the immune system, and thereby carry out its activity of natural cytotoxicity. So it is able to naturally carry out cytotoxicity. So, like mentioned, it has this very simple function, and that is that it is cytotoxic. So, it can lyse cancer cells, it can lyse virus infected cells, it can produce inflammatory cytokines, and therefore you can understand that it is therefore contributing to what is called as protective immunity or innate immunity, with the main objective of protecting the system from a foreign antigen. Now, the molecular mechanism of natural cytotoxicity by natural killer cells involves three main processes. It recognizes the target cell, it will contact, so it will actually form connections with the target cell, and on making connections, there is a signal transduction, and with that signal transduction, it leads to apoptosis of the target cell. So it leads to what is called as a target cell death. So this absolutely is important for the functioning of the natural killer cells. That is, the natural killer cells will have to adhere or will have to make a contact with the target cell. So if this is the membrane of the target cell and this is the membrane of the natural killer cell, you can see it can make n number of interactions. These are different ligands that are present on the natural killer cell and corresponding ligands that are present on the target cell. If these connections are made, then there is a certain kind of signal that passes on to the natural killer cell. Now you can see that some of the interactions are inhibitory. So therefore, if it is inhibitory, the natural killer cells will not kill the target cell. But if the interaction is activating, then the natural killer cell will get activated to kill the target cell. So that is something that is very important. And so they have to differentiate. So the NK cells have to differentiate between a normal cell and a transformed cell. So only if it is a transformed cell, it should kill the transformed cell. But if it's a normal cell, then it should not kill the normal cell. So there has to be a way of distinguishing between the normal cells and the transformed cells. So we will look at it. But then what is also important is that with these interactions, a dynamic equilibrium has to be established. As in how many interactions are inhibitory and how many interactions are activating, and therefore depending on that equilibrium, it is decided whether the natural killer cell will kill or not. So the integration of multiple activating and inhibiting receptors is an important phenomenon with respect to the natural killer cell acting as a cytotoxic molecule. Cell. Now, we did mention that natural killer cells have to identify whether the cell is healthy or it is transformed. So as it has been mentioned that natural killer cells are able to kill tumor cells. So many studies have been done to understand how natural killer cells can distinguish a healthy cell from a transformed cell. So what has been observed is that you can see that if that you have the natural killer cell actually recognizing a healthy cell through the MHC class, that class 1 that is exhibited on any nucleated cell. Any nucleated cell will generally have an MHC class 1. So if you have an activator receptor and an activator ligand binding and an inhibitor receptor and the MHC class 1 binding, then they negate each other. And because they negate each other, the natural killer cell is going to recognize the healthy cell uh, because there is no tilt towards either activating or tilt towards either inhibiting. So there will be no killing of the healthy cell. In many cases where you have the MHC missing, okay, then there are chances that only the activating receptor and the ligand bind, The inhibitory receptor is not binding to anything on the surface of the target cell, and so the equilibrium moves more towards activating the natural killer cells. So the natural killer cells will get activated, and they would be affecting the target cell. Or what you could have is that you have the inhibitory receptor binding to the MHC class 1, but because the cell has got transformed, or because the cell is harboring a virus inside it or a bacteria inside it, and it is on its surface exhibiting certain receptors which can be, or certain ligands which can be recognized by the natural killer cell receptors. So therefore, again, the tilt will be more towards activating the natural killer cell, and so you would have the natural killer cell killing the target cell. So in this case, the target cell itself is kind of, because of the presence of the activating ligand, allowing the natural killer cell to kill the target cell. Now, when one looks at the functions of natural killer cells, the natural killer cells are able to bind to cells which are infected by virus. So you have ligands that can specially bind to surface markers present on virus infected cells, or they can bind to surface markers that are present on tumor cells. And they can also get activated, or get, yes, the natural killer cells can get activated by several cytokines or growth factors or chemokines that are released by other immune cells. So, for example, dendritic cells or macrophages, due to its exposure to a foreign particle or a foreign antigen, may release many cytokines, and these cytokines in turn can activate natural killer cells, and the natural killer cells, on getting activated, can kill the target cell. Similarly, T helper cells and B cells can also through their IL-2 activate natural killer cells, which in turn will give rise to several cytokines like gamma interferon, GM-CSF, CCL3, 4, 5, and these in turn will be able to, get activated and function. It can also release a lot of cytotoxic vesicles, so that these vesicles release the hydrolytic constituents present inside it, or the constituents present that can kill the target cell. So these cytotoxic vesicles will be able to kill the target cell. So what is observed over here is these are the specific, ligands or cytokines that are, sorry, not ligands, but cytokines that are released by the natural killer cells, and that leads to several different functions. So for example, if the natural killer cell is able to give rise to these different cytokines, then it leads to anti-tumor activity. If these are the cytokines, then it is antiviral and bacterial. If these are the cytokines, then they help in help adaptive immunity. So they will help T cells to actually differentiate into into effector T cells and memory T cells to actually give rise to adaptive immunity. It can also help in tissue regeneration. So if there is a tissue damage, it can help in repairing the tissue. It does carry out regulatory functions because it can secrete IL-10, and it can also, carry out adoptive cellular, can be adopted for cellular therapy. So there are a lot of NK cells that are being engineered in such a way that they be used as therapy for cancer. Now, what has been found from very recent studies is that natural killer cells may be able to form memory natural killer cells. And natural killer cells, because they are releasing IL-10, can also have a regulatory function. So it may so happen that when one knows more about natural killer cells, then one may find that the natural killer cells may not just have a role in innate immunity, but it may also have a role in adaptive immunity. However, currently it is not very, very clear. So let us just look at how the natural killer cell functions. So when, suppose, a natural killer cell binds to an virus infected cell, so you can see that this ligand is interacting with this ligand of the infected cell, and because of that there is a signal that is passed on to the natural killer cell, and the natural killer cell releases the vesicles that contain perforin and granzymes. So granzymes are esterases. So perforin will make pores in the infected cell or the target cell, and the granzymes will get into the infected cell and lead to apoptosis. So the granzymes are able to induce cell death in these target cells, and that is how mostly all activated natural killer cells are functioning. But you can understand that a natural killer cell has to get activated to release the cytotoxic vesicles. It will not release unless it is activated. And therefore, this interaction is important. Its ability to identify the target cell is important. Only and only when it identifies a target cell will it be able to carry out the cytotoxic activity. So it does release perforins, granzymes, and it carries out cell death by apoptosis as mentioned. It also exhibits what is called as antibody dependent cytotoxicity. So many times what happens is that if the target cell has certain epitopes to which an antibody, circulating antibody can bind, then the circulating antibody is actually acting as an opsonin. So the Fc component of the antibody can bind to an Fc receptor on the natural killer cell, and therefore the natural killer cell will get activated because of the Fc receptor binding to the Fc of the antibody, and therefore it is called as antibody dependent cytotoxicity, because the antibody, when binding to the natural killer cell, is able to signal the natural killer cell to get activated and carry out cytotoxicity. So let us look at the conclusions. The natural killer cells or killer cells are developed in the bone marrow with several cytokines playing an important role in its maturation. Natural killer cells do not contain T cell receptor and CD3 complex but have several receptors such as killer immunological receptors, etc., that help the cells to recognize target cells. Their cytotoxicity is triggered by recognition and contact of a target cell, leading to release of perforins and granzymes that in turn lead to apoptosis of the target cells. Here it is very important to understand why apoptosis has to be triggered and not just simple lysis of the cell. If it leads to simple lysis of the target cell, then if the target cell is a virus infected cell, then the virus can get released and that virus can infect other cells which are neighboring the target cell. But if it is apoptosis, what is going to happen is that the target cell will definitely get killed along with the viruses present within it. So apoptosis is the mode. Apoptosis is the modus operandi of the natural killer cells. The natural killer cells are also pro-inflammatory because they give rise to several cytokines and they are able to regulate other immune cells, and they also have been reported probably forming memory. So natural killer cells are effector lymphocytes of the innate immune system that controls several types of tumors and microbial infections by limiting their spread. Whose spread? The spread of the microbial infection and the tumor and subsequent tissue damage. Thank you.